大联赛区公开组的决赛马上就要开始。那么进入决赛的2支队伍呢，一支是来自人大附中的队伍，他们做正方会做 first speak team。另一队是来自北京市情国际学校和北京4中的北京11的这个混搭队伍，他们是反方 speak second。那么我们的决赛现在开始。
Pro team, first speaker.
Yeah.
4 minutes, ready when you are.
So, ready? My opponent's ready. My constructive speech starts now. We affirm the resolution that public health services should expand access to gene editing technologies. Observation: according to the WHO, public health service is any organized legal action that helps the public in terms of environment, mental, and bodily functions, including disease prevention. Expanding access is defined as the extended benefit of something. Framework: as long as we prove that gene therapy is feasible, beneficial for humanity, safe to use, and sustainable, we win this debate. We have no obligation to prove anything other than what is presented in this case, because that's not what we're advocating. Contention 1, gene therapy can benefit humans and the ecosystem. Subpoint A, gene therapy can stop disease without modifying genes directly. According to Salk's Institute, gene therapy can use base editing technologies and CRISPR-Cas13D to accurately manipulate the structure of the RNA instead of directly the DNA. Researcher Kornerman claims that as cells build the body, RNA controls protein synthesis. By modifying it, we don't directly harm the cell's DNA. This allows enhanced DNA and proteins to be produced in this natural, less harmful way, using RNA to correct false inscriptions and proteins to kill diseases. Lin MT claims that Alzheimer's, for instance, Parkinson's, and Huntington disease can all be solved by the changes of RNA. CancerNet also claims that with gene editing, cancer-repair genes can cure tumors and allow recipients to remain healthy. Subpoint B, gene editing can reduce the amount of disease that exists. Tangerman from Futurism claims that disease transmitted by organisms can be stopped by modifying genes of vectors. UC Riverside School developed mosquitoes that are susceptible to changes by gene editing, controlling traits that pass to their offspring. By impairing mosquitoes' abilities, the team reduced disease spread to humans, such as dengue and yellow fever. WHO claims that if we hinder these vectors, 200 million people's lives can be saved each year. Moreover, gene editing can cure disease sources such as microbes and bacteria. In 2018, a team of Chinese researchers increased resistance to HIV in mice by replicating a natural gene that prevents disease. As 96% of experiments have succeeded, this method can work on humans. By using base editing to introduce naturally resistant genes to humans that lack it, researchers can bolster HIV resistance. Other institutes have also used similar methods on diseases such as HPV and bird viruses. Subpoint C, gene editing technologies are beneficial to the environment. According to Cornell University 2018, gene editing has allowed bacteria to produce green fuels which allow humans to remain healthy. Other benefits include the reduction of GHG gas emissions from livestock such as cows, pesticide prevention of vegetables without chemical intervention, water efficiency for plants, and even decomposition of garbage through microbes. Contention 2, gene editing technologies are extremely feasible and will be expanded. Subpoint A, gene editing procedures are extremely easy to maneuver. With more professionals delving deep into the domains of gene editing, it has become a safe, innovative solvency for many developments. The success rates for CRISPR is as high as 16 out of every 18 tries. Simple restriction enzymes used to cut 20 base pairs of the DNA can be extremely precise. Though it isn't as fast as CRISPR, Huang SH Technology states that the success rate for base editing is virtually 100% due to it not being subjected to any error by only swapping one base from another. What's more is that more gene editing methods are developed while common methods succeed over 90%. It is more important to note the extreme accuracy of these procedures. Subpoint B, gene editing technologies are feasible and inevitable. According to Metzel U.S. News World Report 2014, gene editing is inevitable. With so many new options to choose from, the costs aren't as high as people think. UC Berkeley states for less than $1,000, thousands of gene editing procedures can be executed. Both developed and developing countries have shown signs of utilization of gene editing on animals and bacteria for beneficial purposes. With Europe, Asia, and the Americans all having success in gene therapy, it is only a matter of time before it spreads even more. Already, China's experts have saved humans' lives with solving genetic diseases. Therefore, we strongly urge a pro-ballot. Thank you.
Thank you. Con team, speaker 1, 4 minutes.
Everyone ready? The con constructive speech starts now. The con team negates the resolution that public health health services should expand access to gene editing technologies. We argue that the status quo already gained the benefits of gene editing technologies without expanding, and to allow any kind of expansion would create severe harm to humanity that is backlashed by the public, which we believe is unacceptable. Observation 1, the status quo. A, gene editing technologies. Medical treatments after birth are not controversial, and germline and embryo genetic editing that are controversial. According to Sarah Reard in 2015, many countries limit research using human embryos. China explicitly bans the implantation of genetically modified embryos in a woman. Despite differences about how far to go in applying gene editing to the unborn, nearly everyone in the meeting agreed that efforts to use gene editing after birth to correct defects in non-reproductive cells is beneficial and should continue. This means that people already have the right to treat diseases or otherwise improve their health standards using somatic editing of genes right now, and there's no need to expand. Condition 1, risks of expanding. Experts warn that we're moving too fast on gene editing. According to Marilyn Macaron, AP News in this year, U.S. health officials are eliminating special regulations for gene therapy experiments. It's an extraordinary milestone for a field that has produced only a few approved treatments so far. This is not the right time to be making any move based on the idea that we know what the risks are, says Stanford Bioethics' Mildred Code. The risks were underscored in 1999 when the team testing a gene experiment put a trio on the field. Cole, who is a member of the RIC, said gene therapies are biologically complex treatments, and we really don't understand how they work. In many cases, there are miraculous recoveries and remissions that we haven't seen before, but there also are very spectacular failures. Condition 2, biotech bubble scenario. Perceptions about development of new drugs and protection advancements lead to huge biotech investments. According to Ireland Waldrop Forbes in 2015, revamping investments in biotechnology on risky stocks inflates the market by injecting too much capital into lower yields. This put investments on the brink. This continued risky investments prolongs the bubble, which will eventually lead to a failure. The impact prolongation will lead to widespread market failure and a longer time frame for recovery. Global health depends on biotech markets. They are the key to prevent diseases and reduce global warming and produce medical treatments. The result will collapse the public health service. Condition 3. Public backlash. A, money has biased decision makers about genetic engineering to avoid restrictions. According to Cameron Scott in 2015, professors at universities gained financial interest in how their discoveries were commercialized. Their commercial interests may have influenced their choice not to consider the question of using CRISPR in agriculture plants and animals at the international meeting, even when the debate about them continued this December. Donut, the inventor of CRISPR, is involved with 3 companies that have already formed in hopes of commercializing CRISPR gene editing. Donut's financial interests in her discovery are more the rule than exception. It will be hard to find someone in the biotech world who doesn't have an interest, because all the principals in this conversation do have what looks like conflicts of interest, said Mercy Damnowski, a PhD. B, lab research on germline editing will create pressure for public use of genetically modified humans. According to the same source, researchers ought to explore how CRISPR works on human embryos. They are arguing that editing human germline cells for research purposes should move forward immediately, while a dialogue about the social and ethical implications of using modified germ cells to initiate a pregnancy is underway. C, backlash against germline editing can hurt somatic editing, which is the kind of technology we have nowadays as well. According to Lanfear et al. In 2015, there is fear of the negative impact it could have on important work involving the use of genome editing technologies in somatic cells. In our view, genome editing in human embryos using current technologies could have unpredictable effects on future generations. This makes it dangerous and ethically unacceptable. Such research could be exploited for non-therapeutic modifications. We are concerned that a public outcry about such an ethical branch will hinder a promising area of therapeutic development, namely making genetic changes that cannot be inherited. And for the following reasons, we believe that we should definitely not expand this technology.
Thank you. 3 minutes crossfire. Both teams ready?
Yeah.
3, 2, 1.
You mentioned about germline editing, right? In your contention 3, backlash against germline editing. If we are not doing germline editing, how does that link to our case?
Well, that's not the point of our contention. Our contention is to provide an evidence that if this technology is too risky and we haven't been fully developed and research is not enough, and we introduce into the public, the result will be a backlash against this technology, and people will oppose this technology, and we will lose when we gain this as well.
However, the card you used is a specific card on germline editing of human embryos. So you may ask a question.
Oh, I can ask.
Yeah.
So do you have any specific examples that RNA succeed on humans?
My partner will explain that further.
You
Yes,
have to
I
answer
do.
it right now.
I
Because
do.
Yeah, you have to.
You'll have to give us time to look for a piece of evidence.
But my partner cannot properly do her rebuttal without.
We're actually doing the rebuttal first, so your partner can't. Okay, thank you. So you mentioned that risks of gene editing are high, right?
Yes.
But do you know there's something called a cost-benefit analysis that we analyze even if there's a risk in a medical procedure? If a person is going to die because there's no other medical treatment of a disease such as diseases we mentioned, then even though it's taking a risk, we have to do it?
We understand that you are saying that if this succeeds, the impact will be very great. But we should also consider if there is the risk, it will cause a backlash, and a backlash will result in we losing those technology, and we cannot afford taking this risk.
May I have a follow-up? So your advocacy is to ban every single technology with a risk,
No,
right?
we're saying that this is not the right time to expand. Given the risks that exist now, we should put more time in this
You have made no
development.
time frame arguments throughout this debate. Please ask the question.
So, you talk about RNA. So how is modifying RNA different from DNA?
It does not change. The DNA is the sequence that defines you as a person. However, when DNA tries to make a protein, what it does is that it transcripts a piece of RNA to transfer this message. And if in this process we edit the RNA in this DNA, we avoid the risks because we are not changing the DNA and we don't do things like mentioned in your card, like accidentally erasing parts of important DNA. And this kind of thing cannot happen if we are using RNA technology. So we're basically doing everything DNA editing can do, but eliminating the risks by using the RNA technology that we mentioned.
We didn't talk about the accident you mentioned.
Okay, but you mentioned the risk. Yeah,
Okay,
that's what. Okay. Another question. So you mentioned that only germline editing is considered expanding access according to your status quo.
The status quo, yeah, well, the point of the status quo is to tell us that we already gain the benefits from somatic cell editing and there's no need to expand.
Okay, so you say, we already have. So have you seen people being edited by RNA editing? Thank you.
Thank you.
We will call for 20 seconds per 100 seconds per time.
20 seconds starts now.
More seconds, please.
Sure. Time.
All right. So, sorry. Judges ready? My opponent's ready? My rebuttal starts now. So first of all, let's go back to the framework. The con side is clearly stated inside their framework that as we have already had the rights to access to certain technologies, therefore we have already been expanding these access. However, I just want to clarify that this framework does not work. Just because we have a certain technology and just because certain countries have used it, this doesn't mean that this has been expanded to the public. Now onto my opposition points. First of all, they claim that it is strictly prohibited to use the germline editing methods, and that certain countries such as China have been using these certain methods to do illegal experiments. Well, my first response to this is that yes, we agree. 29 out of the 39 countries investigated by Japan all have the legal issues with germline editing, and this is why we're not advocating for it. And my second response to their Chinese card is that China did not break any laws while doing this. They did it on the embryos that were going to die. They didn't do it on the babies. They didn't do it on any germline cells. Now onto the contention one. They talked about how about the feasibility and how very few approved methods have worked. But apparently they claimed that this was from the 1999. However, this is 2018, people. We have already seen a 96% success rate in the experiment that we have mentioned in our contention one. Moreover, their evidence is old, which is definitely unreliable. And second of all, when they said that it was difficult, well, I just want to mention, Judge, that we've already determined the sequence for E. coli as well as many other bacteria in the past year. This means that with this new information, we can definitely solve this. Just because taking taking time, it takes time. It doesn't mean that we shouldn't take time in developing new technologies. Onto their contention 2, they talked about how there's a biotech bubble and how that it's very risky, and once it widespreads, the economy could fail. However, Judge, I would like to mention one thing. This is only mentioned inside the private sector. They are only used in these private companies that hold the rights to use these gene editing technologies. However, what we are advocating for is to expand these technologies. This is my second response. If we expand these technologies to the public, that means more people can get the opportunity to solve these diseases, that they aren't locked up in the private sectors that they are mentioning, and that this bubble will be popped indefinitely. We're trying to solve this because we are as concerned as them. However, our method is better than just complaining. Contention 3 about the bias, and how they say that Dodna, as well as the creators of certain of these technologies, are commercialized. Well, first of all, I just want to mention that they've only claimed about the creators of CRISPR. However, there are 7 other technologies out there, and they have made no response to this. My second response to this is that we want this to be commercialized. We want this to expand to the public. We want more people to know about this technology. We want these peoples and these diseases to be solved. We don't want them to be restricted. We want more people to know about this because these technologies are good and cheaper. They also talked about how embryo and germline editing is illegal, and yes, we totally agree with them, but that's not what we're doing. And they also mentioned about somatic editing and how even though we're doing it right now, we shouldn't expand it even more. However, Judge, let me just mention that so many people suffer from cancer. However, they cannot get the help that they need in order to solve these diseases. However, by expanding, we mean the public gets it more. Just because we already have this technology doesn't mean it's expanded. Now onto my case and how we stand out more than our opponents. First of all, they only talked about human editing, all right? They only talked about germline editing and how it's unethical. However, we have the contention that talks about the environment. And according to the National Academy of Sciences, we spend less amount of money trying to save the environment, which is a public health service that benefits the entire society, benefits the entire world for cheaper amounts of money. We do this because we want a bigger impact than our opponents. However, they're only advocating for banning on CRISPR technology because it's somehow illegal, but we already have a higher impact than them. Moreover, according to Larry Gibbleson, people don't trust in these technologies. People fear these technologies because they're not more engaged. What they used to do was they just introduced the technology, but they never explained how it's used. However, according to Larry Gibbleson, as they expanded this technology, more and more people got to know that genetically modified technologies aren't that scary.
Thank you. Fun side. 30 seconds. Starting now. Time.
Okay. Just ready. Opponents ready. Carnivale's speech starts now. Okay, so first I want to talk about topicality. According to our status quo card, currently we are already gaining benefits from our somatic gene editing. So if you're just keeping the current system and somehow giving it to more people, it doesn't mean you're expanding. It just means that you're providing it to more people. It doesn't mean you're providing more branches of it. You're not opening more doors. You're just keeping what it is. And then I want to move on to talk about how the RA experiments cannot be completely safe. The point isn't what kind of technology is used, it's what kind of scientists you use. Already, as my partner has said in her constructive contention 3 card, that scientists are biased by financial purposes. And here are more cards to further that. From Kate Keeland and Ritos, reported from 2008, that scientists in business were underestimating DNA changes. Whatever bad things found out, they hurt their prices. Scientists studying gene editing to a CRISPR-Cas9 have found that it can cause unexpected genetic damage. The findings have safety implications that gene therapy have been developing using CRISPR. They also add to the findings published last month, which suggests that CRISPR may adversely increase cancer risk in some cells. We found that changes in DNA have been seriously underestimated before now. Shares of gene editing companies, CRISPR Therapeutics, Editas Medicine, we do not use the methods described in this natural biological paper. Nevertheless, it is our work. We do not do the similar findings. CRISPR Therapeutics said that Editas
It said that. And EDITA said that company are not specifically concerned about the license finding. That is because if they admit to these disadvantages, it will definitely hurt their prices. So the point that this technology is not safe is because scientists and companies will not be truthful enough to the public to tell them all the risks and problems with the RNA. And moving on to talk about how they're going to edit mice. They say that editing mice who has HIV has already worked, but mice, that doesn't mean it will work for humans. Right now, it is biologically infeasible for humans. Mice don't count. Even knowing what gene to change is a problem that requires substantial investigation. Biology is complicated, and mice and men are not the same organism. Simply showing that edit is effective and saving a model organism is not enough to justify making the change in the same in humans. What could be beneficial to mouse may be harmful to humans. Mice and men are just not the same organism. You cannot apply this argument to humans too. Then moving on to talk about how they say that they're going to edit mosquitoes, and that will somehow prevent stuff from happening to humans. For example, malaria. Editing a mosquito has a problem because editing mosquitoes may have unpredictable effects on the environment. This is according to Victor Tangerima in 2018. But interfering with mosquito populations could have unforeseen consequences. Eliminating a species, even one that doesn't appear to have much ecological value, could upset a careful balance of ecosystem. That could have disastrous consequences, such as disrupting the food web or increasing the risk that diseases like malaria could be spread by different species entirely. So you kill one vector, then what about the other vectors? And furthermore, on more arguments that they're somehow changing creatures and so making the environment better. That is not a part of public health service. The public health service is interested with the health of humans, not the environment around us. That is supposed to be a cons argument. The cons should be concerned with the environment, not pro. And then moving on to them saying that CRISPR is inevitable in the future. CRISPR cannot be inevitable in the future. And gene editing cannot either, because as I said, there are 7 kinds of gene editing techniques, and scientists are separated on developing one of them. So basically, not all scientists are working hard on developing one kind of technology, and they're also incentivized by financial market to not be truthful to the public. So CRISPR is not inevitable, it is not safe, and those other 6 kinds of gene editing technology is not safe either.
Thank you. 3-minute crossfire.
All right.
Second speaker. Both teams ready?
You have the first question.
3, 2, 1.
All right, so if you mentioned about mosquitoes, but let me just recall what happened from my contention. We never said that we were killing it. So if we are only impairing the vision or certain aspects of it, how are we destroying the population? How are we hurting the ecosystem?
What kind of aspects are you hurting?
We're hurting its vision so that it doesn't necessarily see the humans itself. You understand? So that means it doesn't spread.
So how do they eat?
They can eat other animals. They just don't see humans.
Well, if they don't see humans, why can they see other animals?
Because they can.
Well, what is the difference between seeing humans and seeing animals?
Those don't get transplanted into our bodies. Judge, we're saying public health service is implemented on
You
humans.
haven't answered my question.
Yeah. Let me just tell you again, if it spreads to other animals, it's okay. Because the other animals won't spread it to us. A cow won't bite you and give you malaria.
And wouldn't that hurt our environment?
Why would that hurt our environment?
Because your animals are sick.
But they were sick already.
Why were they sick already? They had malaria because you
They had malaria before, but we're already inhibiting one way of them giving malaria to us.
Okay, so you're making more animals to have malaria.
No, not exactly. We're just hindering it from transferring it to
And
humans.
also, if you're hurting the mosquitoes, as my card said, you might make other animals the bacteria instead. So are you going to hinder every animal type?
That doesn't exactly mean we're going to hinder every animal, but that does mean we're stopping it from happening. If you know what I mean.
Then how?
That's still better than what you're saying, just letting mosquitoes bite people and giving them malaria. I really think we should move on to the next question, or else this debate will not be educational.
Okay, sure. It's my. I think I just asked you a question about malaria. So
Okay, yeah. So shouldn't you ask me a question?
Okay, sure. I can ask you a question
Okay,
if you don't have any. Okay, so on your RNA experiments, are there any successful results already?
What do you mean? For
Successful-
what type of where which type of results?
Lab research.
Lab research, yes. Let me just tell you right here. LinMT claims that Alzheimer's, 4 instances of Parkinson's, and Huntington's disease will be solved by the changes of the RNA. Basically what happens is we don't modify the DNA. The RNA produces these proteins that kills off these diseases. that helps the human body. We're not changing the person whatsoever. And if you want to ask for the success rate, it's 96%. We presented it in contention
That's
1.
about mice.
No.
That's about mice and HIV.
Please recall straight back to what we have said in our contention 1. We're saying that by implementing this technology, the success rate is 96%. And if you want to argue even more, in our contention 1, we also said that 16 out of every 18 tries on the humans that have cancer have succeeded.
What about the 2 other tries that failed?
What do you mean?
What if that happens when you're...
Okay, let me just give you a hypothetical situation right now.
And
If there was a
also,
person...
I have my answer to your question, you can ask yours.
Wait, so let me just finish right here. If a person was going to die, and there was a 16 out of 18 chance that you were going to give him a medicine that was cheaper and better than the medicine that you already have, wouldn't you give it to them, or would you just let them die?
But if the 2% happens, other people will die.
So
So
what
you
about
wanna
your
say
medicine
one?
right now? There's a lot of high risk for other sorts of medicine. We have a lower risk already.
We call for a minute for us.
1 minute. Starting now.
Okay.
Speaker 1, 2 minutes, whenever you're ready.
Yeah. Good, ready? My opponent's ready? My summary speech starts now. Okay, so their first argument against us is a topicality argument, saying that we're not expanding active. But Judge, please see this. Do you see people being cured by RNA editing, or do you see mosquitoes being eliminated of their disease-spreading abilities by CRISPR? These things haven't happened yet. So by giving more people the right to access RNA editing and really doing the edit on mosquitoes, we are expanding access, and their definition is somehow flawed. Our opponent's argument on our first contention is that scientists are driven by money and they're all bad. However, this is not true because first, they have only presented arguments on America. However, we have used arguments all around the world, including countries like China. Secondly, this argument makes this debate meaningless. Our opponent says our scientists are being biased by money. But in the same logic, we can say that the scientist that provides evidence for our opponents is also biased by money. And then this debate would become an endless questioning and denial of arguments, and that makes this debate really meaningless. Now to our opponent's points. Our opponents have mentioned a biotech bubble scenario. We have rebutted that in our rebuttal speech by saying that that's the private sector, and we are actually using public health service to guide it. Our opponent's third contention is on the biases, which will cause backlash. However, we have successfully rebutted that argument by saying that the more you educate the public about CRISPR and other gene editing techniques, the less a backlash will happen. And our opponent's first contention is about the high risks. However, their high risks are not linked to our argument about RNA editing, because we are actually using the way that eliminates risks. And about the environment argument also, it is actually said by the American public health care system, directly says that the environment is part of the public health care system. Thank you.
Thank you. On side, speaker 1, 2 minutes.
We have 45 seconds.
Starting now. Time.
So, Jen, ready? What's ready? The con summary speech starts now. So to begin my summary speech, I will identify the biggest clash in this debate, which is whether the benefits of gene editing technology is able to outweigh its risks. The con stance is that we should not expand. First, current technology